know that the organizers of this entire conference were very conscious of the challenge that it would be to keep you awake after lunch. So they decided to talk about regulatory things and hope that that would keep you awake and engaged. So I did study science in college, just across the street, but that's only part of my job now. So this is probably a little bit unique as far as the rest of the talks that have happened so far at the conference. It's regulation specific. So a little bit of regulatory background about biosimilars. So a biosimilar is under the 351K of the law. This is 351A under the Public Health Service Act. And an application under 351A is a standalone program. The standalone BLA, it has to contain all the information and data necessary to prove that it's safe, pure, and potent. We say safe, pure, and potent a lot because, again, that's in the regulations, whereas some of you more familiar with small molecules are more familiar with safe and effective. The Biologics Price and Competition and Innovation Act, or BPCIA, created an abbreviated licensure pathway, and that's how biosimilars came to be. And there can be either biosimilar or interchangeable with an FDA licensed reference product. The reference product would be a 351A application. The abbreviated licensure means that a manufacturer may rely in part on the FDA's previous determination for the reference product. Generally, biosimilar manufacturers don't need as many expensive or lengthy clinical trials, potentially leading to faster access to these products. However, biosimilars and interchangeables are still subject to FDA's rigorous approval standards. So our role is to balance innovation and competition. We recognize that role to help ensure that the U.S. remains a driving force in medical innovation, as well as the importance of robust and timely competition to enhance patient access and reduce cost burdens on patients and our healthcare system. We have and will continue to play a critical role in facilitating increased access to biosimilars. One of the ways we've done that previously is the Biosimilars Action Plan, which was announced in the first half of 2018. It's really kind of a roadmap of what we planned to do to help increase biosimilar access, as well as facilitate biosimilar interactions with applicants and manufacturers on how to move their programs forward. We had some 4 key elements to improve the efficiency in the biosimilar interchangeable product development and approval process, maximize scientific regulatory clarity for the biosimilar product developments, develop effective communication to improve the understanding, and that's not only for applicants, but patients, clinicians, and payers. We wanted to support market competition by reducing gaming of FDA requirements or other attempts to unfairly delay competition. This is a lengthy list of the deliverables that we had associated with the BAP. As you can see, many of these have already happened. We've already released several guidances. The only one that has yet to happen is that last one, and that's coming either this fall or this winter, and that's the enhanced Purple Book. So as of October 1st, there have been 23 biosimilar products that have been approved for 9 reference products. 9 biosimilar products are believed to have been commercially launched. There are 73 programs at the development stage where they're engaged in the biosimilar product development program, where we're able to discuss details of their development, and that's for 38 different reference products. Since the program began in 2012, we've had 30 planned 351K submissions publicly announced from 12 different companies for 9 different reference products. This is a schematic that's quite old at this point, but I wanted to show it to you to show you that a reference product and a biosimilar product are really quite similar, or dare I say highly similar. Brackets are used to show the sites with the minor variations, and we reproduce that with the permission of the EMA. It's their graphic. So the goal of a biosimilar is to establish biosimilarity between their product and the reference product, not to reestablish safe, pure, and potent, or safety and effectiveness. Biosimilar means highly similar to the reference product, notwithstanding minor differences in clinically inactive components. and that there are no clinically meaningful differences between the biological product and the reference product in terms of the safety, purity, or potency of the product. How to demonstrate biosimilarity? The 351 has to include information to show that it's biosimilar, that it uses the same mechanism of action for the proposed conditions of use, but only to the extent that the mechanisms of action are known. As you know, many of these products are highly complex and not always known how their mechanism of action works for each indication. Has the same conditions of use proposed in labeling that have previously been approved for the reference product. Same route of administration, dosage form, and strength as the reference product. And that they're manufactured, processed, packed, or held in a facility that meets the biological product standards for safe, pure, and potent. So this is our totality of the evidence approach. The approval of a biosimilar is based on the totality of evidence. Those first three circles there, analytical studies, animal studies, and a clinical study or studies. The demonstration supports biosimilarity will be based on those three, and the nature of or scope of those clinical studies will depend on any residual uncertainty about the biosimilarity of the two products after conducting structural and functional characterization and any relevant animal studies. We issued the comparative analytical assessments guidance in May of this year. It's a draft guidance that describes recommendations for the design and evaluation of comparative analytical studies. It has considerations for the development of that assessment plan using the stepwise approach I just described to support biosimilarity. It lays out considerations for generating comprehensive comparative analytical data, methods for detecting and characterizing differences between your proposed biosimilar and the reference product, assessment of those differences to understand if they have any impact on clinical performance relative to the reference product, and any comparative analytical data that can inform decisions about the type and amount of animal studies that you, or clinical data that is needed. The new draft guidance provides additional clarity and flexibility for developers on the analytical approaches to evaluating the structure and function, some updated recommendations for collection and analysis of that similarity of data, receptiveness to considering alternative approaches proposed by those sponsors for the analysis of their analytical similarity data, and aspects of the CMC portion of the marketing application. I'm not going that far deep into it because my colleagues that are participating on the panel will also go into more detail. So an interchangeable or interchangeability means that the biosimilar may be substituted for the reference product at the pharmacy without the intervention of the healthcare provider who prescribed the reference product. Interchangeability final guidance was also issued this year, and that provides FDA's current thinking on scientific considerations in demonstrating that a proposed biological product is interchangeable with the reference product. Same thing as a stepwise approach to generate that data, same totality of the evidence approach, and it retains flexibility to provide space for science in this area to evolve and for FDA to provide targeted advice on specific study designs for a particular reference product. Demonstrating interchangeable biosimilarity means that the proposed interchangeable must be biosimilar to the reference product. It also must demonstrate that the proposed interchangeable can be expected to produce the same clinical result as the reference product in any given patient. This likely won't involve additional clinical studies other than those necessary to support other elements of demonstrating interchangeability. And it also, if your product is administered more than once to a patient, you have to show that there's no increased risk for safety events or diminished efficacy due to alternating or switching between the reference product and the interchangeable. Some of those alternating or switching studies will, there may be one or more study, and again, either one or more appropriate conditions of use. That final guidance talks about the switching studies. It outlines considerations, including endpoints, design analysis, study population, conditions of use, and routes of administration that can be studied. You can do an integrated two-part study design, where you start out doing the biosimilarity study and then you extend it to include the switching study. There's an option for sponsors to provide justification to the FDA of why any data would not be necessary And the switching studies are generally not needed for products that are only intended to be administered once. We also mentioned the non-US comparator in the interchangeability final guidance. In the draft guidance, we use the words strongly recommend or strongly recommended the use of a US licensed reference product in switching studies. We no longer say that we strongly recommend, and we include considerations for the type and extent of bridging data needed to justify the use of a non-US licensed comparator. The final guidance also talks about post-marketing data. It clarifies that you can first be licensed as a biosimilar and that data or information supporting licensure can later be referenced to support interchangeability. Post-marketing data from a licensed biosimilar may also be considered when data is necessary. There may be situations where that post-marketing data can reduce uncertainty about interchangeability. Safety and monitoring, this is for all biosimilars and interchangeables. All drugs have risks and benefits. Biosimilars are no different. They can have side effects which are expected to be the same as those of their reference products. As part of our review, we assess the manufacturing process and your strategy to control within product variations. Those control strategies are put in place to help ensure that your process is consistent so that your product can be consistent in the clinical performance area. We do robust post-marketing safety monitoring. It's also an important component in ensuring the safety and effectiveness of all biologics, including biosimilars. So looking ahead for the biosimilars action plan, some of the things that we have still on our plate are the biological product regulatory modernization, a proposed rule for that, and I'll get into that in a second. We need to finalize the reference product exclusivity guidance. We're also working to educate patients and consumers about biosimilars and interchangeables, and awareness of stakeholders seeking additional guidance. So the reg modernization rule that we're working on, the BLA regs were written long before the addition of the 351K pathway, which is the biosimilar pathway. So we're working to update those and modernize them for targeted revisions related to the submission and review of all BLAs. We're also updating them to provide enhanced clarity and regulatory certainty to manufacturers of both originator and biosimilar interchangeable products, and also trying to address any gaming that would prevent or delay competition. The enhanced Purple Book was one of the few BAP deliverables that we have not done yet, but that's coming very soon this fall or this winter. That Purple Book provides a list of biological products licensed under the PHS Act. It will include information about biosimilarity and interchangeability, as well as information about exclusivity. The enhanced Purple Book is in development. It's an improved interface to provide users with a friendly, intuitive interface to provide information about all FDA-approved biologics, and it will hopefully enhance information that's readily available for patients, prescribers, pharmacists, and stakeholders. The BAP action actually said enhancing the Purple Book to include more information about approved biological products, including information relating to reference product exclusivity determinations. We also mentioned the education for patients about biosimilars and interchangeables. We've released several new materials. We're working really hard to reach out to prescribers as well as patients and payers to help them understand what is a biosimilar and how it relates to their reference product. Help them especially understand that these are safe and effective treatment options, that they still go through the same rigorous approval process that we use for our other BLAs. And I'll be happy to take questions at the panel. Yes, thank you.
Our next speaker is Dr. Rachel Novak. She began her tenure